Good morning, and welcome to the inaugural event in the 2020 Focus on Research Seminar Series. My name is Enrica Ziller, Outreach and Events Manager in the Office of Research at the University of Texas at Dallas. I will be your moderator. As we move through today's session, I invite you to post questions into the Q&A box for our guest speaker to answer. Joining me today as host to introduce our speaker is Kelly Palmer, Associate Professor in Biological Sciences. Dr. Palmer, welcome.
Hi, everyone. So I'd like to thank all the attendees for coming to the seminar today. This is really exciting. I'd like to thank the Office of Research for organizing this seminar series. And without further ado, I'm going to introduce our speaker today, who's Dr. Nicole De Nisco. Nicole received her bachelor's and PhD from MIT, where she studied plant microbe symbioses with Dr. Graham Walker. She then did her postdoctoral work at UT Southwestern, where she was a National Institutes of Health T32 fellow. She studied microbial pathogenesis there with Dr. Kim Orth and some collaborators you'll hear about today. In 2018, we were thrilled when she joined the faculty of the Department of Biological Sciences here at UTD. So with that, I'll turn it over to Dr. De Nisco.
Hello, everyone. Thank you for joining me today. I'm excited to be here to tell you about a work that is very close to my heart. And I'd like to thank Dr. Palmer for that wonderful introduction. So today I'm going to be telling you about a serious problem in human health and the approaches that my lab is taking in order to be able to solve it. So the disease I'm going to be talking about is urinary tract infection. Urinary tract infection is a global health issue. It is the most common adult bacterial infection. Over 150 million are reported per year worldwide. And this disease primarily affects women, with over 50% of women experiencing a UTI in their lifetime. The impact of this disease is largely overlooked. And this is in part because it is primarily a women's health issue. And it affects part of our bodies that we are told very often that we should not talk about in public. But I will be talking about this today. So what is UTI? UTI is an infection of the urinary tract by pathogenic bacteria and less often by fungi. It can come in different forms, one of which is uncomplicated UTI. And this can affect either the urethra, where it's called urethritis, or the bladder, where it's called cystitis. Another form of UTI is complicated UTI. And so this often involves catheters or any other abnormality that might explain why a patient might be more susceptible to UTI. When the infection ascends the urinary tract into the kidneys, it is called pyelonephritis. And this type of disease can be very serious as it can develop into urosepsis, which has over a 40% mortality rate. The last kind of UTI that I'm going to talk about is going to be recurrent UTI, and this will be the focus of today's seminar. So what is recurrent UTI? Recurrent UTI is defined as 2 UTIs in 6 months or 3 UTIs in 12 months. And it can be illustrated as shown in this graph as a sinusoidal wave that consists of an active UTI followed by a negative intervening culture and then relapse into rUTI again, usually within a period of 3 months. The symptoms of this disease are pelvic pain, frequency, urgency, and foul odor of the urine. In premenopausal women, the rate of rUTI is actually pretty high. So about 25% of UTIs in premenopausal women will progress into recurrent UTI. This amount doubles when we talk about postmenopausal women. In postmenopausal women, over 50% of UTIs will develop into recurrent UTI. And these cycles of relapse and remission and relapse can go on for years. And as you can imagine, this severely diminishes quality of life. So who are the culprits of this disease? What is causing these infections? In order of prevalence, we have an organism that all of you may have heard of. It's called Escherichia coli, or what we call the uropathogenic version, ZPak. Other organisms that can cause this are Enterococcus faecalis, Klebsiella pneumoniae, and diverse other bacterial and fungal species. Some of the bacterial ones are listed here. So what makes these bugs so bad? Why are they so difficult to kill when they've invaded our urinary tract? Well, one reason is because they are able to colonize and actually invade the bladder wall. And so they can form communities hidden within our bladders that in which they are not susceptible to antibiotics, and they're able to hide from our immune system. So these communities have been seen both in mice, so the image on the left, the green bacterial community was observed in a mouse, and we have actually seen this in human patients with recurrent UTI and the image on the right. So the problem that clinicians face with rUTI is that they're only usually able to manage this disease and not completely cure it. And so the tool they use to manage this disease is antimicrobial therapy. This is the most widely prescribed treatment for rUTI. And they prescribe this treatment in two main modalities. Either low-dose prolonged therapy, so women are taking antibiotics prophylactically for long periods of time, or on self-start intermittent therapy, where women are taking antibiotics as they feel symptoms. And unfortunately, there are very high rates of resistance and allergy to frontline antibiotics that are used to treat UTI. So these therapies are often ineffective. And so a survey of antibiotic resistance and allergy was actually done by Malik et al. In 2017 in postmenopausal women with rUTI. And here are the resistance rates and allergy rates that they found. And what you want to look at are the orange pie slices, and those represent the percentage of women that they surveyed that were sensitive, their infections were sensitive to these three different frontline drugs, TMP-SMX, fluoroquinolone, and nitrofurantoin. And TMP-SMX, which is a sulfa drug, is by far the most commonly used drug to treat UTIs. And as you can see, only 23% of women actually responded or their infections were sensitive to TMP-SMX therapy. So this drug is actually not very useful anymore, especially in this patient population. And the failure of our current therapies to treat UTI has actually made the news. So this is an article from the New York Times where they're talking about how as the infections, UTIs, become increasingly resistant to antibiotics, some standard treatments, like treatments with sulfa drugs, no longer work for this ailment that was once easily cured. And so you may think, well, this is an easy fix. All we have to do is develop more antibiotics. Unfortunately, this isn't happening. The development of new antibiotics is dwindling. And this is most easily visualized by looking at the development of new classes of antibiotics. So these are antibiotics that have a new mechanism of function. And as you can see on this graphic, since the 1990s, no new classes of antibiotics have been discovered. Actually, since 1984, no new classes have been discovered. This is a huge gap in innovation. So where can we look for therapies for recurrent UTI? One thing I want you to remember from this talk is that infection is not a one-sided conversation. So bacteria employ all sorts of factors, colonization factors, cytotoxins, and mechanisms to evade the immune system in order to be able to colonize the host. But the host responds to this infection. We have our innate immune system and our adaptive immune system, and we even have a resident microbiota that's able to provide resistance to infection. And this is important to remember because I told you that 50% of women will experience an rUTI in their lifetime, but this means that 50% of women will not. And so what about their host environment protects them against infection? It's not that they're not being exposed to the pathogens in the environment. There's something about their body that is able to provide resistance to infection. So the question that my lab seeks to answer is, are there aspects of the host response we can target in developing alternate therapeutics for rUTI? And so one aspect of this response is inflammation. And we know already that this inflammation plays a role in recurrent UTI. So on the left, you will see a healthy female bladder. This is observed via cystoscope, so a little camera that's put up your urethra into your bladder. And you can see that the lining of the bladder is smooth, and this is very healthy. Now, on the right, we can see the bladder of a rUTI patient. Now you can see this looks a lot different. You can see all of those little pustules and inflamed areas that are very indicative of the inflammation that we see in rUTI. This is the cystitis that we see, the inflammation of the bladder. We also know that the microbiome itself is a critical host defense and might be an interesting host pathway to target. So the microbiome can actually confer protection against all stages of pathogen invasion. It's able to employ colonization resistance to disrupt the establishment and growth of pathogens within the host. It's able to contain pathogens to prevent their spread into other tissues or body sites, which might be important in thinking about having a UTI, an uncomplicated UTI, progress into pyelonephritis and urosepsis. It can also provide disease tolerance by suppressing the negative effects that pathogens have on the host. So for example, the toxins that pathogens excrete. The resident microbiota can actually help us tolerate these toxins better. So when I think about recurrent UTI, I don't like to think about it as a one-sided conversation. It's not just the bacteria infecting the host. I actually like to think about it as a vicious cycle. So recurrent UTI can be seen as a vicious cycle that's initiated by infection. The host immune response to this infection produces excessive inflammation that results in tissue damage. Infection and inflammation can both displace or alter the resident microbiota, and this is called dysbiosis. And although this hasn't been characterized specifically in recurrent UTI yet, it's been observed in other inflammatory diseases that have a microbiota interface, like IBD. So currently, we only have one strategy to break the cycle at a single point, and that's the point of infection, and that strategy is antibiotics. So we have antibiotics that can hopefully block or stop the infection, right? But when we're thinking about developing new treatment strategies, we actually must focus on breaking the cycle at other points. So breaking the cycle at the dysbiosis caused in the microbiome, or breaking the cycle at maybe modulating the inflammatory response to the infection so that it's more positive and less damaging. And so I'm going to tell you about 2 short stories about how my lab, or strategies my lab, is taking to target both the inflammatory response and the microbiome as potential therapeutics for recurrent UTI. So the first story is about identifying targets for immunomodulatory therapies for recurrent UTI. And so this project takes advantage of the knowledge that we have gained from mouse models that that has outlined the Cox2 pathway. And so this is a specific inflammatory pathway that's involved in a lot of different diseases that may be a promising target for immunomodulatory therapies for recurrent UTI. So work in mice has identified this pathway as a key mediator of recurrent UTI susceptibility. And they found that mice with high levels of Cox2 during primary infection are actually sensitized to chronic and recurrent infection. And the inhibition of the Cox2 pathway with selective inhibitors can actually prevent recurrent UTI in mice, which was a pretty exciting finding. And what's important is that we already have FDA-approved Cox2 inhibitors that are widely used in the population to treat ailments such as rheumatoid arthritis. So we already have a drug that could possibly target this pathway. But the question is does this pathway actually play a role in human UTI and recurrent UTI? We can't take at face value the mouse models. They are good to provide direction, but we have to show that it can translate to a human host. So when we were thinking about evaluating this pathway in human patients, we looked at the pathway and saw that the product of the Cox2 enzyme, PGE2, might be a good biomarker to look at. This, this molecule is actually secreted in the urine and is very easy to measure by ELISA. So perhaps we can measure the level of Cox2 pathway activity by measuring the level of its product, PGE2, in the urine in a non-invasive manner, and determine if this might be a potential rUTI biomarker. So the goal of this project is to evaluate urinary PGE2 as a biomarker for recurrent UTI in humans. And so all this work is done in collaboration with our clinical collaborator at UT Southwestern, urologist Dr. Philippe Zimmer. So we developed a study for postmenopausal women in which the inclusion criteria were, of course, that they were postmenopausal, that they had uncomplicated rUTIs, so there's no catheter involved or no abnormality of the urinary tract. These women did not have a pelvic floor procedure within 6 months, and they had no other type of surgery within 1 month. And also they were immunosufficient, so no immunodeficiency, no cancer or chemotherapy being experienced by these women. So we sorted the women into 3 cohorts, and we're going to actually use these same cohorts in the next story, so pay attention. The first cohort is our Never UTI cohort, and this consists of women who have no clinical history of a UTI in their lifetime. Our second cohort is our Remittent rUTI cohort. These are women who have a history of rUTI, but do not currently have an infection. And then finally, our Relapse rUTI cohort are women with a history of rUTI that have a current infection at the time of their office visit. So at the time of their office visit, we collected their urine by clean catch method, and we measured PGE2 by ELISA. And what we found was that urinary PGE2 was elevated during active UTI. And this work was done by a team of students led by one of my graduate students, Tamina. And so what we found was that the median PGE2 levels were twofold higher in relapse versus never UTI cohorts. And this urinary PGE2 was also higher in relapse versus remittent cohorts. The difference was 2.2-fold here. Interestingly, there was no change in the never UTI versus remittent cohorts, which suggests that perhaps this elevation of PGE2 is not a long-term effect. It's not something that happens and is sustained even after infection has resolved. But taken together, these data suggest that urinary PGE2 may be a biomarker for active rUTI, so not resolved rUTI, but active rUTI in postmenopausal women specifically. So we went on to look to see what effect diabetes status might have. And the reason we did this is because rUTI is highly prevalent among diabetic individuals. And a biomarker that is not confounded by diabetes would be very useful to clinicians. So we separated patients into new groups based upon their current infection status and whether or not they had diabetes. And this, just for reference, is all type 2 diabetes. And the trend that we saw of elevated PGE2 during relapse was maintained in diabetic individuals just as in non-diabetic individuals. So this suggests that PGE2 is an effective biomarker for recurrent UTI independent of diabetes status and could be very useful for clinicians. We took this story one step further, and we wanted to understand if urinary PGE2 levels could predict rUTI relapse in our cohort of women. So we followed the patients in the relapse cohort for 1 year. And we recorded the time to rUTI relapse for each patient, and this was done blinded. We separated patients into two groups, above and below median PGE2 levels. And we found that patients with above median PGE2 levels at their time of first infection relapse more quickly than those with below median levels. And the hazard ratio of this was actually 0.277, which is much higher than we were expecting. And so this means that the hazard of relapse was about 3.6 times greater for the above median group than for the below median groups. So this is a pretty strong effect that we're seeing. So what conclusions can we take from this first part? The first conclusion is that urinary PGE2 is a biomarker for active recurrent UTI and is predictive of rUTI relapse in our specific cohort of postmenopausal women. So one question you might have, especially if you're suffering from UTI in the audience, is does this mean I should be taking Cox2 inhibitors for my UTI? My answer to you is no. We have not gotten there yet. The next step is a blinded, placebo-controlled, randomized clinical trial. This is the only way we will be able to tell if Cox2 inhibitors are an effective therapy for recurrent UTI. One thing we're also interested in doing is developing a rapid test for urinary PGE2 that clinicians can use in office. And this is important because we want clinicians to be able to select women that might benefit from Cox2 inhibitor therapy if it turns out that it is effective, because Cox2 inhibitors can carry some side effects with them. So I'm going to switch gears completely, and I'm going to tell you about the second side of my lab, which is focusing on the microbiome in order to develop microbiome-based therapies for recurrent UTI. And so before I get started telling you about this, I need to mention that urine is not sterile. And this might be contrary to what you have been told in the past. but a urinary microbiome exists. And I like to illustrate this by showing you these plates, these agar plates that we use to grow bacteria in the lab. And on these plates, we have plated patient urine. And we have plated patient urine from patients that have UTIs and from patients that are completely healthy. And I don't think you would be able to predict which plates belong to which patients. The plates outlined in red here are actually from patients that are completely healthy and do not have a current infection. And as you can see, there are plenty of bacteria within their urine. So urinary microbiome definitely exists, and this has been proven in both culture and culture-independent methods. So what do we know currently about the urinary microbiome? One major limitation is that only the urinary microbiome of premenopausal women has been characterized. And so this microbiome has been characterized primarily by a method that can only detect which genera are there and has been separated very preliminarily into different urotypes. And one thing they've noticed about the premenopausal microbiome is that it's low diversity. So it's dominated by one or two major species. So the urotypes that they've identified so far in the premenopausal microbiome are Lactobacillus. So this is believed to be protective genera, Gardnerella, Corynebacterium, Streptococcus, and Staphylococcus. But the biological significance of these urotypes is completely unknown. It's still a very, very new field. And so the goal of our project here was to gain mechanistic knowledge of the urinary microbiome that's required for the development of microbiome-based therapeutics. And so the method we choose to employ for this was a method called whole genome metagenomics. And the reason we chose this method was because it has the potential to provide insight into microbiome function. And so what whole genome metagenomics can do is it can tell you not only who is there. So most of the studies that have been done on the urinary microbiome have been using techniques that only tell you who is there. This technique can tell you not only who is there, but what they might be doing, because it can tell you all the genes that that community is encoding. And then from those genes, you can infer the proteins that are being made and how they might be doing all of these functions. And so the workflow for this kind of analysis is actually pretty simple. All we do is take the urine that we've collected by the clean catch method. We isolate the genomic DNA. It's fragmented, put on a very fancy sequencer called an Illumina sequencer. And analysis and many different types of analysis are performed. But basically, we get the sequences of all the genes that are present in that urine sample. And so we're able to run different kinds of analyses on these genes to understand the phylogeny, so who is there, but also to do gene-based functional profiling to understand what functions these different bacterial species may encode. And so before I get too far into the data, I want to tell you about the team that is working on this project. It's actually a very large project and requires a very cohesive team. So we, of course, have our clinical collaborator, Dr. Zimmern, without whom we would have no patient samples. Dr. Palmer is my close collaborator, and we are working together on all the metagenomics. Dr. Chao Xing is at UT Southwestern, and he is the lead in the bioinformatics on the project. And then we also have a new collaborator, Dr. Kiwei Lee, in the Department of Math at UT Dallas, and he's doing all the mathematical modeling of our data because we have some really big data sets, and he's employing some mathematical models so we can understand them better. And this work was really spearheaded by a very talented ex-graduate student in our lab, now Dr. Michael Nugent, who will hopefully be joining myself and Dr. Palmer as a postdoc soon. So we developed this same cohort design that we employed in the PG2 story, where we had cohort 1 was our never UTI, cohort 2 was our remittent rUTI, and cohort 3 was our relapsed rUTI. We applied a little different inclusion criteria here because we were measuring something different. We were measuring the microbiome. So we still have our postmenopausal women with uncomplicated rUTI. But in this study, we left out diabetic patients. And this is because we don't know the effect of diabetes on, the microbiome. And we also left out women that had taken any antibiotics within the past 4 weeks, because as you can imagine, this might affect the composition of the urinary microbiome. And the same other inclusion criteria where they didn't have any pelvic floor procedures and that they were immunosufficient. And so here are our summary statistics for the cohort of women that we studied. So we had 25 women in each cohort. They all had a similar average age, BMI, and urine pH. And so our metagenomics results, right now we have pretty much done just the taxonomic analysis, as even this is quite computationally expensive. We have detected 373 total microbial species belonging to 162 genera across all patients. Here in this heat map, I'm showing you the top 26 genera that we detected. And so the bright blue squares mean 100% abundance, and the black squares mean 0% abundance. And you can see that it's all based upon the scale bar there at the bottom. And we got a lot of information, very useful information in this analysis, but I just wanted to point out 2 things because I'm only going to tell you one short observation that we made. And that was that we saw that the genera Lactobacillus was enriched in the never UTI and remittent rUTI cohorts. So you can see outlined in red here, we have the genera Lactobacillus, and you see more of those blue and close to blue colored squares in the never UTI and remittent UTI groups than you see in all the patients that were in the relapse. And then similarly, and this was completely expected, of course, because this is our uropathogen here, E. coli, we saw that the genus Escherichia was enriched in our relapsed rUTI patients. And this was just completely expected. But what I wanted to focus on was this observation with Lactobacillus. And the reason is because we went into this analysis with a hypothesis that Lactobacillus, which everyone thinks is this protective genera, would be more enriched in this never UTI cohort than even in the remittent rUTI cohort, because we thought that it was affecting the susceptibility to recurrent UTI. And as you can see by looking at these 2 side by side, just visually, this was not the case. We can look at this graphically, and I can show you that there is no significant difference in abundance of Lactobacillus between the never UTI and the remittent rUTI cohorts. So we were a little disappointed with this result because, of course, it did not support our hypothesis, but that's science. And so we looked a little further into the clinical metadata, and we noticed a trend with patients taking vaginal or taking estrogen hormone therapy. So estrogen hormone therapy is common in postmenopausal women to treat the symptoms of menopause. And so when we separated our women about not by their UTI status, but by if they were taking estrogen hormone therapy or not, we actually found that now there was a very strong association of Lactobacillus abundance with patients taking estrogen hormone therapy. It was a very interesting result, but we were a little perplexed because we could see this kind of bimodality here of patients where we saw this very high enrichment of Lactobacillus, and then some patients taking estrogen hormone therapy that didn't really show very much enrichment at all. And so what we did is we actually divided our patients further into groups by the type of estrogen hormone therapy they were taking. And so the women in our study were taking 3 different types of estrogen hormone therapy: oral, patch, and vaginal. And surprisingly, we found that this association with lactobacillus abundance was lost in the women taking vaginal estrogen hormone therapy. And only women taking oral and patch had this strong association with lactobacillus abundance. And this goes against common clinical belief that vaginally applied estrogen hormone therapy would be more efficacious at encouraging lactobacillus colonization. So what conclusions can we take from this part of the talk? The first is that use of estrogen hormone therapy, or EHT, is associated with an enrichment of bacteria in the genera Lactobacillus. And we actually did some further mathematical modeling with Dr. Kiwei Lee, and we're able to show that specifically this was in the species Lactobacillus crispatus. And this is important because L. crispatus protects against bacterial vaginosis, so it's a different disease, and certain sexually transmitted infections. But its role in recurrent UTI is completely unknown, and what it's doing in the urinary tract has not been studied. One really important observation was that the modality of EHT may be important, and surprisingly, the weakest association with lactobacillus abundance was seen with vaginal EHT. And so this raises a new question: why might vaginally applied EHT be less effective? And so one possible explanation can be dosage or compliance. And so we're seeking to test if this might be the explanation by measuring the amount of estrogen in the urine of these patients by mass spectrometry in collaboration with Dr. Vladimir Shaleyev at UNT. Another possibility is that there might be members of the urinary or even the vaginal microbiome in these individuals that are actually actively degrading the estrogens in vaginal estrogen therapy. And this is another line of investigation that we will be following. And of course, one thing we really want to know, we really want to figure out, is what is the function of the urinary microbiome? We know a lot about the function of the gut microbiome. We know that the gut microbiome allows us to digest our food, is important in providing colonization resistance, but we know next to nothing about the function of the urinary microbiome. And so since we have this large data set with this potential for functional insight, we are currently performing functional analysis that will hopefully be completed very soon. And to supplement this, we are also doing metatranscriptomics, so that will allow us to look at the gene expression profiles of these microbiomes, so we can not only see what genes they have, but what genes they might be expressing. And then metabolomics, so we can look specifically at the metabolism that is involved in each of these different types of microbiomes. So with that, I'd like to thank my lab and all of my collaborators. Specifically, I'd like to thank Dr. Michael Nugent. Sorry, I forgot the doctor there. He just defended a few weeks ago. And then Neha Huliakar, who helped him with some of the metagenomic stuff. And then Tamina, she is the one that spearheaded the PGE2 project. And of course, my wonderful collaborators, Dr. Kelly Palmer, Dr. Kiwi Lee, Philippe Zimmern, of course, and Dr. Chao Xing, and our funding sources. So with that, I will take questions.
Wonderful. Thank you, Dr. De Nisco. We do have a few questions in the chat box, and I invite everyone listening to go ahead and post more if you have them. The first one is, from the causes of bacteria listed that invade the bladder, where do these bacteria come from?
That's a really good question. So there are kind of two hypotheses for this. One is that they are coming from the gut. right? So that they're just existing as reservoirs in our gut, and then they're moving into our urinary tract through contamination from that area. Another hypothesis is that, especially in these women that have this recurrent UTI, is that the bacteria that invade the bladder are actually able to kind of hang out there and are actually kind of reinitiated and reactivated and cause infection directly from reservoirs within the bladder itself. Or even some groups are finding that we can find these bacteria in the vaginal epithelium, which is super interesting.
Okay, so nothing to do necessarily with body soap, laundry soap, city water?
No.
No. Okay. Do you have any holistic recommendations for treating chronic UTIs?
Well, I am not a physician, so I cannot give recommendations of those sorts. I think the best non-antibiotic therapy that I think even most clinicians will prescribe is estrogen hormone therapy. There have been some trials that have shown its efficacy, but it's been really interesting because those trials have had mixed results, and that's, I think, might be because all the women were in those trials were actually using vaginal estrogen hormone therapy. So we think that either because of compliance or dosage, or maybe there's this crazy microbiota that can degrade that estrogen before it can have its effect, is not effective. So we're hoping to understand more about that by looking at the different modalities of treatment.
Okay. And you mentioned the lactobacillus. So we see that a lot advertised in taking yogurt. So is that one way to up our dosage of that, or bioavailability for it?
So the lactobacillus in yogurt is there are actually species of lactobacillus that are present in your gut, which are different than the ones present in your vagina. So so those have not been proven to be very helpful. I mean, no species of lactobacillus has been completely proven to be helpful with recurrent UTI. The trials on that have been mixed too, and I think it's because you need to combine estrogen and probiotics, but the specific types of lactobacillus in yogurts are not the ones that are, thought to be protective in the genitourinary tract.
Okay. And what about drinking cranberry juice? That kind of goes along with the holistic, right?
Yeah, so I don't think the scientific evidence on that is very strong. But if it makes you feel better, sure. I'm not a physician.
Can you maybe go back over why you focus so much on the postmenopausal women, why you think they were more susceptible to UTIs?
So we focus on postmenopausal women because they're an underserved demographic. So most of the research in this field has been done in premenopausal women and has been done using like juvenile mouse models. And so we see that there is a higher incidence of this disease in postmenopausal women. So our rationale is that this is a group of women that might benefit the most from the insights that we can garner from this. And also we think that there's something really important about hormone status and susceptibility. And if we can kind of work out that major difference, and it's a starting point, right? So it's not that we don't care, we care a lot about premenopausal women with recurrent UTI, but this is a starting point for us in a population that really needs our help.
Excellent. And then I'm not sure how much you know about the women that were in the study, but how different were the women that the samples were taken from? For instance, were they of different ethnic backgrounds?
Yeah, so they were reflective of the patient population at UT Southwestern in the urology clinic. So We did not have the luxury of trying to do any kind of enrichment in different groups. So it was primarily Caucasian women, and that's one of the limitations of the study. And we definitely would like to expand our studies in the future to include more ethnically diverse groups. But this was just, representative of the demographic in that clinic.
Excellent. Just so you know, we have lots of questions coming in. This is fantastic. Thank you, everyone. So we've got a thank you for incredibly informative presentation. I understand that your research focuses on the host side of the equation. If we were to focus on the bacteria and their ability to develop colonies and communicate between colonies to enhance the issue through quorum sensing, would that also be a way to tackle the issue? And would there be more complications by focusing on this side of the matter?
So, I mean, there are, we actually do study bacterial virulence in my lab, but that's, I didn't want to get into that. But yeah, I think there are a lot of different ways that we can target even the pathogen. And there's been a lot of talk about antibiotics that are not kind of aimed at killing the pathogen, because the bacteria resistance is going to be selected for more strongly if you have an agent that is you need to be resistant to this or you're going to die, than if you had an agent that kind of just was reducing the virulence, so reducing the ability of that bacteria, kind of like turning it into something that was more of a commensal, that was more of a good bacteria. So kind of taking away its weapons, right? And so there are a lot of labs that are focused on kind of developing therapies that can disarm pathogens, and I think those might be promising as well.
Okay. And how much PGE2 in the urine is from the rest of the body from other inflammatory conditions?
And so so that was one of our exclusion criteria. Also was one of them was taking these. So we didn't have any women in the study that had like rheumatoid arthritis or lupus or things like that that might confound this. And so and you can see very clearly the association. We did a bunch of analysis and tried to associate it with all these different factors, and the only factor that we found this really strong association with elevated levels was having an active infection. And that's because the response of this pathway to infection is so strong, it's so robust. And then that's something that's kind of just known from looking at the responses in mice and other things.
Okay. In addition to not taking diabetes patients, how else were you controlling for external variables that might affect the data?
And so this is in our microbiome study, I'm assuming, because that's the one where we so we had a pretty strict exclusion criteria. So it was just uncomplicated UTI. It's women in a very tight kind of age group that are all the same menopausal status. And anything that could really affect the function of their urinary tract, we had as an exclusion criteria. So, and that's kind of included in this umbrella term of uncomplicated rUTI. So any women that had like a high post-void residual, so this means that they're retaining a lot of urine in their bladder, or women that had prolapse, these women were all excluded. And so the women really needed to be otherwise, healthy. And so that's why we had, and I think our study is one of the ones with the most strict exclusion criteria that we've at least seen published so far. So but there is going to be inherent variability, and we did power analysis and we determined what cohort size we would need to be able to see a certain effect size, and et cetera. So we did take that into account, and you always have to balance the availability of subjects and the kind of effect sizes that you're aiming for.
Okay, thank you. Let's see. Are you or other research groups investigating recolonizing infected bladders with good bacteria, similar to a gut microbiota transplant?
Yeah, so that's one of our thoughts, is developing kind of a rationally developed probiotic therapy. So this would be probably in the form of a suppository in the vagina. And so one of the things that I didn't mention is that the vaginal microbiome and the urinary microbiome are actually very interconnected. And so one of the ideas is if we can restore the lactobacillus levels in the vagina, then that will kind of translate into the urinary tract and can help restore health there too. So that's also that's definitely something we're looking at. But as we're doing further analysis, we are seeing that, you know, it might not just be enough to give suppository of lactobacillus. We might have to come up with a specific consortium of bacteria that can help each other thrive, or we might need to provide it with estrogen or something like that that can really help them colonize. So that's one of the challenges, is getting the bacteria to colonize once you introduce them.
All right, thank you. And I know you mentioned a few antibacterial treatments used for UTIs. ZPak is also a common treatment for most bacterial ailments. Is this equally ineffective?
I'm not sure. I don't think so UTIs, I don't think are deemed important enough to use ZPak on very regularly, but at least with the clinicians that we work with, the 3 frontline drugs are really that TMP-SMX, different fluoroquinolones, which have their own issues of toxicity, and then nitrofurantoin, which is by far the most effective antimicrobial, and that's because of its mechanism of action. It actually concentrates in the bladder, and so it's very specific for the bladder, but it also has toxicity, which might be a problem for some women.
Indeed. The next question is, how do you prevent cross-contamination with the vaginal microbiome in the clean catch urine technique?
So so we actually are not as worried about that kind of contamination, because again, we have this linkage between the vaginal and the urinary microbiota. So we are really specifically looking at the bacteria that are in the urine, right? And so the women do, of course, they wipe themselves with a sterile wipe before they give the urine sample, and they, you know, make every effort to not contaminate. But every different sampling method has its pros and cons, and if we were to collect by catheterization, there are you know, potential contaminating effects there, and then the only other method to really get just bladder resident bacteria is something called suprapubic aspiration, and I don't really think that it's very invasive. It's basically putting a needle into someone's bladder and aspirating out the urine. This is done from like above diaphragm, and it's just not. I don't think it would not be reasonable for this kind of study at all. It's too invasive, in my opinion.
Okay, thank you. For your sample sizes, would you like to have larger numbers if possible? And if so, how large would that ideal number be?
I think it depends on the experiment, but we've actually been able to make some really interesting observations that I didn't have time to talk about today with our current cohort sizes, and we are collaborating with Dr. Lee, who's actually applying some new methods of mathematical modeling that actually allow you to get more insights from smaller sample size data by removing different confounders and things like that. So yeah, but I mean, of course, you know, we would love to double our sample sizes if that were possible, but you have to weigh that to how expensive these experiments are to perform. The sequencing is not cheap, so if I had infinite money and infinite time, then sure, doubling it would be great, but I think we can make a lot of really interesting conclusions just from what we have.
Okay. And then our next question shares that my mother's UTI episodes are accompanied by cognitive effects, such as confusion. In fact, confusion is a strong predictor of UTI. Is anything mechanistically known about the connection to cognitive function?
No, there's no mechanistic knowledge of that, but you know, I think there's so much talk between the brain and different parts of the body that we just don't understand. But also just the pain that's associated with these. I mean, these infections are incredibly painful. Like if you haven't had one, then you don't realize how painful they are. They're painful. And so that in itself, being constantly in pain can definitely, might translate to a cognitive effect. But yeah, that's an area that definitely needs to be studied.
Okay. What causes a change in the urinary biome for postmenopausal women? Would controlling factors that lead to this change reduce this cohort of women to have increased risk of UTI?
So we really think that, one of the really clear-cut things is that the incidence of rUTI in premenopausal women is almost half of that as in postmenopausal women, or at least the incidence of UTI developing into rUTI. So we really think that hormone status has a large effect on it, and that hormones play a really important role in helping our microbiome, making sure that the right bacteria are colonizing our vagina and our urinary tract, and are able to provide some kind of colonization resistance against. And then also it could, completely do with age-associated immune decline, because at the same time these patients are older, and as we know, our immune system gets less effective as we age. So that might also have an effect. And so that's why we're kind of looking into both of those sides of the equation.
And we've got just a couple more minutes before we end. We've got just one more question in the queue. What Cox2 inhibitor drugs are already available on the market?
So the big one is celecoxib. And so that one is what's currently used to treat like rheumatoid arthritis. I think it's Celebrex is the brand name maybe. And so this drug, it is widely used, but it does especially, it carries side effects like all drugs are. So our goal is really to kind of develop a more personalized approach to treating this infection. And so like our kind of dream is if you go into the clinic and the clinician can measure the PGE2 levels in your urine, and if it's above a certain threshold, then they might suggest that you undergo this therapy in adjunct with antibiotics to kind of reduce your chance, hopefully, of getting recurrent UTI. And that's kind of our dream, but we have a lot of work to do to get there.
Okay. Oh, I think we've got time for just one more that came through. What treatment path are you hoping to eventually develop? Are you looking to correct urinary biomes or develop new medication treatment to treat the growth of harmful UTI-inducing bacteria?
So our lab is currently focused on kind of restoring the host response right now. So that is primarily through the immunomodulatory therapies like the Cox2 inhibitors, and then probiotic, and we're imagining estrogen combination therapies. But we really have to do more work to understand which bacteria need to be included in that probiotic to really make sure they're able to colonize and everything like that. So that's what we're focusing on now. But there's a lot of other groups that are focusing on that other side of the question.
Excellent. Well, thank you so much for sharing your time, talent, and resources, Dr. De Nisco. Did you have any other closing remarks?
No, just thank you all for coming, and I really appreciate your attendance and wanting to learn more about this.
Thank you. Everyone, join us 2 weeks from today as we welcome chemistry PhD candidate Juan Pablo Viziduet to the second 2020 Focus on Research seminar on May 29th. Please see the announcement in the Q&A section for the link to view the complete seminar schedule and to register. Thanks again, everyone, and have a great afternoon.